Hello everyone. So the next topic which we will be discussing here is about the denaturation. So what is denaturation? What is the definition of the denaturation? So the definition of the denaturation is the denaturation implies the destruction of the tertiary structure of a protein molecule, and it will lead to the formation of a polypeptide chain. Now, the protein has generally four structures. So what are the four structures? They are primary, they are secondary, then tertiary and quaternary. So we are talking about the tertiary structure here. So what are we talking about? We are talking about the tertiary structure here, that denaturation is the destruction of the tertiary structure of the protein molecule and the formation of a random polypeptide chain. So denaturation of a protein is one of the phenomenon that will result in the disturbance of the stability and the structure of the protein. So as you can see in this particular figure, there is a folded protein which was being there, but after the process of denitrification, that will turn into a unfolded protein. So the chemistry of protein has always been important to the biomolecules in the living system. So the fundamental blocks of our body structure and their functioning require the protein. So this protein is supplied to our body through the food products such as the pulses, cheese, milk, nuts, etc. Now, as you can see in this particular figure, there was a denaturation process which occurs. So these biomolecules. So what causes the denaturation of proteins? So the biomolecules are also required for the proper maintenance of our body. So there were three structures I have already told you: the primary, tertiary, secondary, tertiary, and quaternary. The stability of the protein and its structure depends upon the physical and the chemical conditions. So it totally depends upon the physical and the chemical conditions. Now, the denaturation process basically of proteins, it is caused by the temperature, mainly by the temperature and the pH, which affect the stability to a greater extent. So first I will talk about is the temperature. So temperature, there are high levels of thermal energy which disrupts the hydrogen bonds that hold the protein together. So when these bonds are broken, the proteins will begin to unfold and they lose its capacity to function as intended. So the temperature at which the protein denature may vary. So that will vary, but most common human proteins function optimally at the body temperature, which is near to about 37 degrees of Celsius. So that is what is the temperature. The next is the pH. So pH also plays an important role in changing the structure of the protein. So this is a figure in which there is a both groups that are being protonated, which is having a positive charge. After that, there was a deuterion, which there is a neutral charge, and after that, both the groups, they are deprotonated. So this is a diagram which is only showing that pH also plays a important role in the changing of the structure of the protein. So due to the changes in the temperature or in the pH or other chemical activities, the hydrogen bonds they present in the proteins get disturbed, and this results in the unfolding of the globular proteins and leads to the uncoiling of the helix structure. So the uncoiling of the helix structure affects the chemistry of the proteins and they will lose their biological activity. So this phenomenon of losing their activity and uncoiling the helix structure due to physical or chemical changes, it is known as the it is known as the denaturation of the proteins. So during the denaturation of the proteins, the primary and tertiary structures get destroyed and only the primary structure is being retained. So the covalent bonds, they are broken, and there is an interaction between amino acid chains which gets disrupted. So this results in the loss of the biological activity of the protein. So they next some of the examples which include the denaturation of the protein. So this figure is representing that only. So it is often possible to reverse the denaturation process because the primary structure of the polypeptide, the covalent bonds holding the amino acids in the correct sequence, is intact. So once the denaturation agent is removed, the original interaction between the amino acids return the protein to its original conformation and it can resume its function. So in the first figure, there is a egg and example of egg which is being taken. So what is happening is, so denaturation can be irreversible in extreme situations like frying an egg. So the heat from a pan, it denatures the albumin protein in the liquid egg white and it becomes insoluble. So the protein in meat also denatures and becomes firm when cooked. So this is the cooked one, and this is when there is a frying of an egg. So the next figure is about the denaturation is occasionally irreversible, in which the protein albumin in raw and cooked egg is being shown. The next is about the paperclip. So a paperclip analog which visualizes the process when cross-linked. Paper clips, they no longer move freely, their structure is rearranged and denatured. So this was about the example of the denaturation of the proteins. Now some other examples as well. So for example, heating, exposure to acids or bases, or even physical action can cause the denaturation to occur. So heavy metal poisons such as lead and cadmium, they change the structure of the protein by binding to functional group on the protein structure. So denaturation of the protein can be done by bringing in the physical changes as well as the introduction of the chemicals. So some of the common cases of denaturation of the protein are coagulation of an egg white when egg is subjected to boiling, and denaturation occurs due to the change in the temperature. Curdling of milk is another example of denaturation of protein where the formation of lactic acid by microbial action results in the process of the denaturation. So this was all about the denaturation. These are some of the references. Thank you.